Hello. हेलो एवरीवन। मेरा आवाज़ आ रहा है आप लोगों को? So today we will discuss 100 important or golden points related with the FMGE exam. Okay. Now these are completely based on previous year topics or previous year questions. There are separate FMGE video on Meded channel which covers these PYQs in detail. Okay. Which covers these PYQs in detail. There are two videos, FMGE1 and FMGE2. Today we will discuss the important points related with these PYQs, previous year questions or previous year topics. And one by one, you can note these 100 golden points related with these PYQs. Okay, so let's start. See, I am not reading all the questions here. We'll be dealing with the important points related with the PYQs. Now see, whenever you see the condition that is diarrhea, dementia and dermatitis. See, 3 Ds, diarrhea, dementia and dermatitis, and if untreated, kept untreated, it can lead to the 4th D, that is death. So niacin deficiency causes diarrhea, dementia and dermatitis. Niacin, B3, vitamin B3, this is the first important point. अगर आप लोगों ने ये थोड़ा सा पैटर्न एफएमजी या कोई भी एंट्रेंस एग्जाम का पैटर्न थोड़ा सा स्टडी किया, द टॉपिक्स विच आर रिलेटेड विद द क्लिनिकल डिसीजेस आर फ्रीक्वेंटली आस्क्ड इन द एग्जामिनेशन. So try to recollect these points from any book. Okay. So diarrhea, dementia, dermatitis causes niacin deficiency. We'll be discussing near about 95 or more questions here. So it is very difficult to cover each and every minute detail of that topic related to that PYQ, but we'll try to recollect the things and try to remember the things. Okay. So diarrhea, dementia, dermatitis, it is due to niacin deficiency. Now, second important point is what are the causes, means how this topic is interlinked with the other topic. Which other topic it is linked? It is linked with the tryptophan metabolism. This niacin, it is linked with the tryptophan metabolism. Okay, so the factors or diseases which affect the tryptophan metabolism also causes the niacin deficiency. So it can be due to the deficiency in the diet. Hartnupp's disease means defective absorption of tryptophan and other neutral amino acids. Defective absorption of tryptophan and other neutral amino acids. Carcinoid syndrome or argentaphinoma. It is a tumor of argentaphin cells of the intestine in which tryptophan, more than 60% of tryptophan, it is diverted for the serotonin production. So tryptophan, it is converted into serotonin. So it will be not available for the niacin synthesis. So there is deficiency of niacin. Pyridoxal phosphate deficiency, which is particularly required for the kynureninase enzyme. Pyridoxal phosphate deficiency in tryptophan metabolism, it is particularly required for the kynureninase enzyme. And if there is deficiency of pyridoxal phosphate, the xanthurenate 3-hydroxykynurenine, it is converted into xanthurenate and it is excreted in urine. In vitamin B6 deficiency, this is also a golden point. Okay, in PLP deficiency, there is xanthurenate excretion in urine, that we will discuss in the PLP. Also the drug, the anti-tubercular drug isoniazid causes pyridoxal phosphate deficiency. Okay, so and the people eating maize and jowar as a staple diet, okay, because niacin in maize, it is present in bound form, so it is unavailable for absorption, and in jowar, because of high leucine content, it inhibit the enzyme qprtase, which is an important enzyme in the tryptophan to niacin conversion. Okay, so apart from this, the tryptophan will give the important products. Tryptophan will give you, apart from this, is both glucogenic and ketogenic amino acid. Aromatic जो amino acids है, they are both glucogenic and ketogenic. So tryptophan will give you glucose, tryptophan will give you fat, tryptophan will be incorporated into a proteins. Okay, apart from this, there are specialized product given by the tryptophan, that is NAD, NADP, serotonin, melatonin, and it is excreted as hydroxyindole acetic acid. So this is related with the tryptophan. Here golden points are diarrhea, dementia, dermatitis. Dermatitis specifically involves the sun exposed part of the body, specifically the neck, and the exposed part of the hand and foot, that gives glow and stalking appearance of the hand and foot, and in the neck it is Castle's necklace dermatitis. Okay, this is third important point, that it causes Castle's necklace dermatitis. So you may get a image based question on this part. So niacin and thiamine, these are the very important vitamins on which there is the question is frequently asked. Okay, so these are the 3 points related to this. Also these are the specialized product given by the tryptophan. Okay, so what is answer to this question? It is niacin, diarrhea, dementia, dermatitis, vitamin B3. Okay, see again, diarrhea, skin rashes, dementia. Which of the vitamin deficiency is most likely cause? It is vitamin B3, that is niacin deficiency. Now see, this is Castle's necklace dermatitis. Dermatitis involving the sun exposed part of the body. These questions are discussed in 2 separate videos for FMGE exam. Okay, now today we will revise the important points related with this PYQs. So this is Castle's necklace dermatitis. It is a feature of niacin deficiency. Also it gives it affect the sun exposed part of hand and foot, gives glow and stalking appearance of hand and foot. See, this is the glow and stalking appearance of hand and foot. Now the second topic, now we are jumping to the next topic, which is related with the pyridoxal phosphate. We already discussed pyridoxal phosphate. The INH therapy means isoniazide therapy, which is an anti-tubercular drug. When prescribed, it causes deficiency of pyridoxine. Okay, it causes pyridoxine deficiency. Now, what is cystathionine beta synthase? Cystathionine beta synthase, it is an enzyme which is required for the transsulfuration reaction in the metabolism of sulfur containing amino acid. What are the sulfur containing amino acid? Methionine, cysteine and cysteine. These are the sulfur containing amino acids. Methionine, cysteine and cysteine. In methionine and cysteine metabolism, the cystathionine beta synthase, it causes the conversion of, now see this metabolism, homocystinuria is a disease which is related with sulfur containing amino acid metabolism. It is due to the defect in cystathionine beta synthase. Okay, this is pyridoxal phosphate dependent enzyme. It causes conversion of homocysteine to cystathionine and cystathionine to cysteine. Means it is important in the synthesis of cysteine. And remember this reaction, these are very important reactions, because this is a transmethylation reaction in which S-adenosyl methionine is a universal methyl group donor. So SAM is a universal methyl group donor. This is important golden point. Universal methyl group donor for transmethylation reaction. Yes, adenosyl methionine. And universal sulfate donor is phosphoadenosine phosphosulfate. This is universal sulfate donor. Okay, this is methyl group donor in the transmethylation reaction. Can you recollect some transmethylation reaction? Norepinephrine is converted into epinephrine. Goniodoacetate is converted into creatine. Okay, epinephrine is converted into metanephrine. N-acetyl serotonin is converted into melatonin. These are the transmethylation reaction. So methyl group which is donated by S-adenosyl methionine, it is accepted by an acceptor and it is converted into the methylated products. Suppose norepinephrine is converted into epinephrine. So this is transmethylation reaction. Then S-adenosyl homocysteine is converted into homocysteine. Homocysteine is converted into methionine. And this is very, very important reaction because this reaction is connected with vitamin B12 and folic acid. It is connected with vitamin B12 and folic acid and it is known as folate trap. It is known as folate trap. देखो होमोसिस्टीन यूरिया इंपॉर्टेंट है, विटामिन B12 भी इंपॉर्टेंट है, फोलिक एसिड भी इंपॉर्टेंट है और सैम भी इंपॉर्टेंट है. यस, एडिनोसिल मिथ्योनिन. Now what is folate trap? What is functional folate deficiency? Functional मतलब है लेकिन कुछ काम का नहीं है. That is known as functional folate deficiency. Now this is metabolism of sulfur containing amino acid in which methionine is converted into homocysteine. Homocysteine is converted into cysteine. Okay. Now what is folate trap? This reaction conversion of homocysteine to methionine is catalyzed by methionine synthase. It is catalyzed by methionine synthase and for this reaction it requires vitamin B12. It requires vitamin B12 and in this reaction methyl tetrahydrofolate is converted into tetrahydrofolate. So this is B12, this is folic acid. Now whenever there is deficiency of B12, it block the conversion of this methyl tetrahydrofolate and to tetrahydrofolate and it get trapped as methyl tetrahydrofolate and there is no other use other than this reaction. There is no other use of methyl tetrahydrofolate in our body. So this tetrahydrofolate is blocked as methyl tetrahydrofolate, that is known as functional folate deficiency. That's why in B12 and folate both deficiency there is megaloblastic anemia because it is related with DNA and RNA synthesis and as the nucleus size decreases because decreased synthesis of DNA and RNA and cell growth is arrested, means cell cycle is arrested in the S phase of cell division. So cell grows continuously without a cell division. So there is larger cytoplasm. So macrocytosis, megaloblastic anemia. So these are the important points related here. So this is cystathionine beta synthase. Defect in this enzyme. This is pyridoxal phosphate dependent enzyme. When it is blocked, there is increased level of homocysteine which causes the cardiovascular episodes. It is responsible for thromboembolic episodes, homocysteine, because it causes the generation of reactive oxygen species on reacting with the collagen molecule. So it causes oxidized LDL formation and atherosclerosis. So homocysteine, elevated level of homocysteine is there. As homocysteine is elevated and this reaction is blocked, methionine levels are also raised. So methionine homocysteine level elevated in homocysteine urea and cysteine level decreases. So this should be restricted in the diet. Methionine, homocysteine and cysteine should be supplied in the diet in patient with homocysteine urea. The typical characteristic feature of homocysteine urea is ectopia lentis, means subluxation of lens, and the patient or the child appears like a Marfan syndrome, means the long digit archanodactyly. These are the features of the Marfan syndrome. So point number 4, homocysteine urea, it is due to defect in cystathionine beta synthase. Then point number 5, vitamin B12 deficiency also causes the folate deficiency. Point number 6, SAM is the universal methyl donor, and phosphoadenosine phosphosulfate is a universal sulfate donor. Okay. Here answer is cysteine. Now see, macrocytic anemia means megaloblastic anemia with increased excretion of methyl malonic acid. As far as biochemistry is concerned, there are 3 important causes of megaloblastic anemia. First one is the folate deficiency, second is the B12 deficiency, and third one is orotic acid urea, which is connected with pyrimidine metabolism. कौन से 3 बोले मैंने, recollect करो. First one is the folate deficiency, second one is the, just wait, so first one is the folate deficiency, second one is the B12 deficiency, and third one is the orotic aciduria, which is connected with the pyrimidine metabolism. Now see, if there is megaloblastic anemia along with increased level of methyl malonic acid in the urine, increased level of methyl malonic acid in the urine, it is due to B12 deficiency. There are 2 important reactions catalyzed by the B12. Which are the 2 important reactions catalyzed by the B12? One is the methionine synthase reaction, and second one is the methyl malonyl CoA mutase reaction. Okay. So whenever there is blocked in the methyl malonyl CoA mutase, coenzyme A mutase reaction, this methyl malonic acid is elevated and it excreted in the urine. So elevated methyl malonic acid is there whenever there is B12 deficiency. Folate has 5 important functions in our body. Folate, it is important for C2 and C8 of the purine synthesis. Folate is required for the thymidylate synthesis. It is required for conversion of glycine into serine. Then it is important in the histidine load test in which FIGLU is harm aminoglutamic acid. FIGLU is excreted in the urine. Okay. So these are the important reactions catalyzed by the folic acid. Folic acid, as it is involved in the DNA and RNA synthesis, the deficiency causes the megaloblastic anemia. Okay, whenever there is FIGLU excretion in the urine in the histidine load test, it is due to the folate deficiency. Folate deficiency in pregnancy causes neural tube defect in the child or fetus. So megaloblastic anemia with increased excretion of methyl malonic acid, it is seen in deficiency of B12. Okay. Now see, these are the important features of the megaloblastic anemia. When you called it as megaloblastic anemia, when size is much more, increase in mean corpuscular volume. So when the size is much more, RBC size is much more, that is macrocytosis, that is, and hypersegmented neutrophils, that indicates megaloblastic anemia. So megaloblastic anemia with increased FIGLU excretion on histidine load test is a feature of folate deficiency. Okay, the next important point, see, this is comparison normal blood smear and megaloblastic anemia, the large oval RBCs and hypersegmented neutrophil. This is a blood picture of megaloblastic anemia. So we already discussed there are 3 important causes of megaloblastic anemia: B12 deficiency, folate deficiency, and orotic aciduria. B12 deficiency, folate deficiency, and orotic aciduria. Vegetarian patient. Okay, vegetarian means taking folic acid. Foliage means leaves. It is present in the leaves. Means if it is taking vegetarian patient present with anemia, if this patient is vegetarian, means it is lacking the B12, because B12 it is present in the non-vegetarian diet. Non-vegetarian diet. Okay. See, Now the subacute combined degeneration of spinal cord is a feature of vitamin B12 deficiency. Why there is subacute combined degeneration of spinal cord? Because of decreased myelin synthesis and defective myelin synthesis. Decreased and defective myelin synthesis. We already discussed, this is a point number 11, in which we already discussed that methylmalonic acid level increases in B12 deficiency. See, this is a B12 role. It causes the conversion of propionyl coenzyme A to methylmalonyl coenzyme A, and methylmalonyl coenzyme A is converted into succinyl coenzyme A. So this reaction, methylmalonyl coenzyme A mutase, it is catalyzed by the B12. It is catalyzed by the B12. If there is deficiency of B12, this is accumulated and excreted in urine. Whenever there is increased level of methylmalonic acid, okay, it competes with the malonyl coenzyme A, and it also incorporates into glycolipids and sphingolipids that causes the defective myelin synthesis. As it competes with the malonyl coenzyme A, it inhibits the fatty acid synthesis. So there is decreased synthesis of fatty acid, decreased myelin synthesis, and also there is defective myelin synthesis that causes degeneration of spinal cord in the B12 deficiency. Okay, so this is important point. Now vitamin B12 is found maximum in. Now remember some dietary sources, some important dietary sources which is a feature of that particular vitamin. Now see, thiamine, it is present in outer alluron layer of the cereals. जो चावल गेहूं हम लोग खाते हैं उसके आउटर लेयर में रहता है. जैसे चावल आप लोगों ने देखा होगा, जब चावल उगता है तो उसके ऊपर जो लेयर रहती है, वी रिमूव दैट लेयर. आफ्टर दैट रिमूविंग दैट लेयर in meal, there is the process known as polishing of rice. So polishing of rice removes the 80% of the thiamine. Thiamine is present in outer alluron layer of the cereal. If excessive polishing is done, it removes the thiamine, 80% of the thiamine activity. Vitamin A, it is present in cod liver oil, shark liver oil, and beta carotene, it is present in orange color fruits, that is carrot, papaya, mango, etc. Vitamin E, it is present in the vegetable oils. Vitamin D, it is sunlight, which causes the conversion of 7-dehydrocholesterol into cholecalciferol. Vitamin B12, it is present in the non-vegetarian sources, while folic acid, foliage means leaves, it is present in the vegetable sources. So these are the dietary sources of these important vitamins. In rest of the vitamin, you can directly, if there is question, you can directly go for you can write anything, but these are the special features of these vitamins, special sources of these vitamins. So the micronutrient that is deficient after ileal resection, ileum. So remember the three important absorption, that is iron, folic acid, and vitamin B12. Just wait. See, iron, folic acid, vitamin B12. It is DJI. Iron, folic acid. In pregnancy, we supplement iron and folic acid. So iron, folic acid, then B12. So iron from the duodenum, folic acid from the jejunum, and B12 from the ileum. This is a site for absorption of these vitamins and minerals. Iron is a mineral. Okay, so iron is absorbed from the duodenum, folic acid, it is from the proximal jejunum, and B12 from the ileum. These are the predominant site of absorption of these mineral and vitamins. Now see the point number 14, ओरोटेट एक्सक्रीशन. हमने बोला था ओरोटेट कब ओरोटेट एक्सक्रीशन मीन्स ओरोटिक एसिड यूरिया. ओरोटेट मीन्स ओरोटिक एसिड यूरिया. तो whenever there is orotic aciduria, it also causes megaloblastic anemia, because orotate, it is related with the pyrimidine nucleotide synthesis. It is related with pyrimidine nucleotide synthesis. In these are the intermediate steps in which orotate is converted into OMP, that is orotatidine monophosphate, and OMP is converted into UMP, that is uridine monophosphate. So these are the deficient enzymes which causes orotic acid urea, that is OPRTase and OMP decarboxylase, O2. OPRTase, orotate phosphoribosyl transferase, and OMP decarboxylase. These are the these enzymes are deficient in the orotic aciduria. So in orotic aciduria, there is megaloblastic anemia. Orotic aciduria is a disorder related with pyrimidine nucleotide synthesis, not degradation. Pyrimidine nucleotide synthesis. In orotic aciduria, there is megaloblastic anemia, failure to thrive, and the excretion of orotate crystals in the urine. Apart from orotic aciduria, one disease is there in which orotate excretion in urine occur, that is type 2 urea cycle disorder. Type 2 urea cycle disorder. Due to, that is type hyperammonemia type 2, ornithine transcarbamylase defect, there is excretion of orotate in the urine. Okay. So this is orotic aciduria. The deficient enzymes are OPRTase and OMP decarboxylase. Now this is a typical picture of self mutilating activity. हम लोग भी थोड़ा बहुत नाखून खाते हैं, सब मैक्सिमम लोग तो नाखून खाते हैं, स्पेसिफिकली जब इंडिया पाकिस्तान का मैच चल रहा रहता है ना, तो और जिन लोगों ने बैटिंग किया रहता वो तो खाते ही खाते हैं, ठीक है, थोड़ा बहुत नॉर्मल है, लेकिन सी द फिंगर्स, सी द नेल्स, ओके, एग्रेसिव बिहेवियर एंड self mutilating activity typically affecting the male child. It typically affects the male child. See the case. Mother came to OPD with 14-year-old mentally retarded boy complaining of joint pain and irresistible urge to bite his finger and lips. Irresistible urge to bite his finger and lips. The serum uric acid level was found to be elevated. Mention the defect. This is Lesch-Nyhan syndrome. Lesch-Nyhan syndrome, it is a X-linked disorder. It is a disorder of purine salvage pathway. There are 2 to 3 important pathways for the purine nucleotide synthesis. One is de novo synthesis and second one is the purine salvage pathway, in which the nitrogen bases adenine, guanine, the nitrogen bases hypoxanthine, these bases are directly converted into nucleotide IMP, AMP, GMP. That is known as purine salvage pathway, and this pathway is particularly important in the brain because brain is dependent on this pathway for the nucleotide synthesis. As there is decreased production of nucleotide due to the defect in the purine salvage pathway and due to the defect in the enzyme, that is hypoxanthine guanine phosphoribosyl transferase, it causes the conversion of hypoxanthine to IMP and the guanine to GMP. Because of decreased nucleotide synthesis in the brain, it causes neurological manifestation in the form of this irresistible mental retardation and irresistible urge to bite their finger and lips. Okay, and there is increased uric acid level because of defect in the HGPRTase, there is increased level of PRPP, which is diverted for the de novo synthesis of nucleotide, which increases the uric acid level. Okay, so Lesch-Nyhan syndrome, remember, whenever there is irresistible urge to bite their finger and lips along with mental retardation and increased uric acid level with a male child, that indicates the Lesch-Nyhan syndrome. तो यूरिक एसिड से रिलेटेड कितने डिसीजेस है बायोकेमिस्ट्री में, अगर सोचा तो 2 इंपॉर्टेंट डिसीजेस है, वैसे तो 3 इंपॉर्टेंट डिसीजेस है. यूरिक एसिड से रिलेटेड। जब भी यूरिक एसिड आएगा 3 डिसीज के बारे में सोचना। कौन से 3 डिसीजेस है? सबसे पहले तो हमको इसके बारे में सोचना है, गाउट। उसका तो टिपिकल फीचर रहेगा, फर्स्ट मेटाटार्सो फाइलेंजियल जॉइंट इज इन्वॉल्व। ओके, सेकंड इंपॉर्टेंट रहेगा, सेकंड इज लेश निहान सिंड्रोम, second important disease in which uric acid level increases. And third important disease is bone Gauts disease, that is glycogen storage disorder. This is the third disease in which uric acid level increases. Okay, so uric acid level elevated along with this feature indicate the Lesch-Nyhan syndrome due to the defect in the HGPRTs. Got it? Now the gout. Gout important case. गाउट का मतलब क्या होता है? गाउट इज ए inflammation of first metatarsophalangeal joint. This joint is mainly involved in the gout, pain in the great toe, and particularly the episode it is precipitate when it is a chronic alcoholic. Alcohol precipitate the attack of gout because alcohol shift the metabolism, or alcohol causes the conversion of monosodium urate into uric acid. It causes the conversion of monosodium urate into uric acid, and this is 10 times more soluble as compared to the uric acid. Uric acid is less soluble, more precipitable, that causes the symptoms or aggravate the symptoms of gout. So gout, there is increased uric acid level. There are primary and secondary causes of the gout. When it is related with the de novo fatty acid synthesis, de novo synthesis of purine nucleotide, these are the primary causes that include in PRPP synthetase activity, increased activity of PRPP synthetase, increased activity of glutamyl amidotransferase, then Lesch-Nyhan syndrome, that is HGPRTase defect. This is decrease, the increase HGPRTase defect. Then von Gauss disease, that is glucose 6 phosphatase defect, and glutathione, increased activity of glutathione reductase. These are the causes of primary gout, while secondary gout includes lymphoma, leukemia, cancer, renal failure, etc. Just remember this: increased activity of PRPP synthetase, increased activity of PRPP glutamyl amidotransferase, and increased activity of glutathione reductase, and decreased activity of HGPRTase, that is Lesch-Nyhan syndrome, and decreased activity of glucose 6 phosphatase, that is von Gauss disease, in which uric acid level is increased. Okay, test, it is the aspiration and microscopy. Okay, needle aspiration of the joint, synovial fluid aspiration and microscopy, in which negatively birefringent crystals are seen. So microscopy, aspiration and microscopy, this is a method, gold standard method for diagnosis of gout. Okay, and which drug is given in the treatment of gout? It is allopurinol. Okay, so this is a gout. This is the toughest deposit, crystals of uric acid deposited in the joint, that causes pain, that causes inflammation, swelling, and deposits in the joint. Okay, so this is a drug which is given in the treatment. So this is the 17th golden point, that is allopurinol is a drug which is given in the treatment of gout. It is competitive as well as suicide inhibitor of xanthine oxidase enzyme. This is xanthine oxidase enzyme. It is competitive as well as suicide inhibitor. Are you getting me? It is competitive as well as suicide inhibitor of xanthine oxidase enzyme. So how it will act? It is a competitive inhibitor. It structurally resembles the hypoxanthine. There is competition between these two, the substrate hypoxanthine and the allopurinol. They compete for the active site of an enzyme, xanthine oxidase. This is known as competitive inhibition. And what is suicide inhibition here? This allopurinol is converted into alloxanthine. Allopurinol is converted into alloxanthine and it is a more potent inhibitor of xanthine oxidase than the allopurinol. And this conversion is caused by, ये conversion कौन करता है? Xanthine oxidase. That's why it is also known as suicide inhibition. मतलब इसको इनिबिट कर रहा, किसको इनिबिट कर रहा? अलोजैंथिन किसको इनिबिट कर रहा? xanthine oxidase को. ये एंजाइम को इनिबिट कर रहा, xanthine oxidase को. और इसको बना कौन रहा? xanthine oxidase ही बना रहा. So that's why it is known as suicide inhibition. So it is competitive as well as suicide inhibitor of xanthine oxidase. This is a drug which is used. This is a nucleotide drug, synthetic nucleotide analog. It is used in the treatment of gout. Okay. On which enzyme will it work? The answer is xanthine oxidase. Okay. Now the different things related with the Km and Vmax. So don't see, just remember there is nothing like increased Vmax. Increased Vmax मतलब मैक्सिमम वेलोसिटी के ऊपर, ऐसा कोई कुछ नहीं रहता कि मैक्सिमम वेलोसिटी के ऊपर कोई जा नहीं सकता. जैसे हमारे गाड़ी में अगर स्पीडोमीटर है, उसकी एक मैक्सिमम लिमिट है, उसके ऊपर हम कैसे जाएंगे? तो देर इज नथिंग लाइक इंक्रीज वी मैक्स. तो अगर आपको ये ऑप्शन में दिखता है तो जस्ट ट्राइक आउट दैट ऑप्शन. इंक्रीज वी मैक्स कभी नहीं रहेगा. ठीक है. नाउ रिमेंबर दिस ट्रिक्स. See, whenever there is X-shaped graph, if it is a image based question, this is line viewer Burke plot. Whenever there is X-shaped graph, it indicates competitive inhibition. X-shaped graph, okay, competitive inhibition. When it is a V-shaped graph, see, this is V-shaped graph, it is a non-competitive inhibition. V-shaped graph, it is non-competitive inhibition. And when it is a, when there are parallel lines, it is a uncompetitive inhibition. So X shape, V shape and parallel line. Competitive inhibition, X shape graph, non-competitive V shape graph, and competitive inhibition X shape graph, and uncompetitive inhibition parallel lines are there. Now see, in competitive inhibition, Km increases and Vmax remains same. Vmax remain same. And in non-competitive inhibition, Km remains same and Vmax get lowered. Vmax get lowered. Okay. In uncompetitive inhibition, both decreases. Km decreases, Vmax decreases. Okay. So try to remember this. In competitive inhibition, there is competition. If you go on increasing the substrate concentration, that will replace the inhibitor. So that will increases the velocity of the reaction. So Km increases. What is Km? Is nothing but the substrate concentration. If you increases the substrate concentration, the velocity will increase. So Km means substrate concentration. So in competitive inhibition, the Km increases, while in non-competitive inhibition, Km remains same, Vmax decreases. See the graphs. Okay, this is competitive inhibition X-shaped graph. This point indicates the 1 by Vmax. Okay, so Vmax remain constant and Km, in case of competitive inhibition, Km increases. Okay. Non-competitive inhibition, Km remains same and Vmax, this is 1 by Vmax get lowered. And parallel lines both decreases in the uncompetitive inhibition. So this is example point number 21, gamma carbox 20, first the 20. So identify the drug Z marked by the arrow. This is Z marked by the arrow. This is vitamin K reductase. Vitamin K reductase. Now see, there are two drugs which act here. So one is the dicumarol and second one is the warfarin, oral anticoagulant drug. Dicumarol and warfarin, these are the competitive inhibitor of vitamin K reductase, epoxide reductase, vitamin K epoxide reductase. कौन से दो ड्रग? ओरल एंटीकोएगुलेंट, warfarin and dicumarol. Okay, dicumarol and warfarin, these are the oral anticoagulant drug that inhibits the enzyme vitamin K epoxide reductase. Okay, and 21st point, gamma carboxylation. What is the function of vitamin K? Vitamin K, there are three forms, K1, K2 and K3. Three forms are there, phylloquinone, menaquinone and menadione. Menadione is synthetic water soluble. Remember A, D, E, K, these are the fat soluble vitamins. But K3, menadione, is a synthetic water soluble vitamin. Synthetic water soluble vitamin. K3, K1, K2, K3, phylloquinone, menaquinone, menadione. विटामिन के का क्या फंक्शन है? विटामिन K1 काय में रहता है? ग्रीन लीफी वेजिटेबल, पर्टिकुलरली स्पिनच. K2, it is intestinal bacteria, produced by the intestinal bacteria, and K3, it is a synthetic water soluble form. And what is important function of vitamin K? It causes the activation of clotting factor 2, 7, 9, 10. 2, 7, 9, 10, activation of clotting factors. How it activates the clotting factor? C, it causes the gamma carboxylation of glutamic acid. Gamma carboxylation of glutamic acid. So there is one negative carboxyl group in the glutamate. It causes gamma carboxylation. Again, one carboxyl group is added. It gives negative charge to that gamma to that glutamate, gamma carboxyglutamate. It carries additional negative charge with which combines with the calcium, and it is important for activation of this clotting factor 2, 7, 9 and 10. So it is important vitamin in the coagulation process. K for coagulation. K start with the coagulation in German. Coagulation starts with the K in German. Okay, so it causes, this is a MCQ, it causes activation of factor 2, 7, 9, 10, protein C, protein S, and protein S, and it causes osteocalcine, activation of osteocalcine. So this is role of vitamin K. This dicumarol and warfarin, these are oral anticoagulant drug that inhibit the vitamin K epoxide reductase. This is 21st important point. 21st important point. Okay, so try to remember this. Statin, this is very common example, statin. And also one that is malonate, which inhibit the succinate dehydrogenase. This is also a common example of competitive inhibition. Statins group of drug, these are prescribed. जैसे आप लोगों के घर में कोई बुजुर्ग आदमी होगा तो उनकी टैबलेट्स वगैरह चेक करना, जिनको हार्ट की बीमारी है, तो उनके इसमें लोवेस्टैटिन, सिमवेस्टैटिन, अटोरवेस्टैटिन नाम के ड्रग्स उनको दिए गए रहते. ठीक है, दिस ड्रग डिक्रीजेस द कोलेस्ट्रॉल लेवल. So this is through multiple step it will give cholesterol. So this is intermediate in the cholesterol synthesis. Starting point is acetyl coenzyme A. Okay. So this is a step, rate limiting step in the cholesterol synthesis, that is HMG, hydroxymethyl glutaryl coenzyme A reductase. This is a rate limiting step in the cholesterol synthesis. And statins, these are competitive inhibitor of this HMG CoA reductase. They compete with हाइड्रोक्सी मिथिल ग्लूटारिल कोएंजाइम. ये सबस्ट्रेट है, ये इनिबिटर है, ये एंजाइम है. ये दोनों इसके एक्टिव साइट के लिए कॉम्पीट करते हैं. And it decreases, statins decreases the cholesterol level by inhibiting the HMG CoA reductase enzyme. So statins group of drug, they inhibit the enzyme HMG coenzyme A reductase. So here answer is HMG coenzyme A reductase. Now sodium fluoride present in the fluoride bulb is an inhibitor of, it is an inhibitor of enolase. This is practical aspect. While collecting the blood सैंपल फॉर द ग्लूकोज, जब भी ग्लूकोज का एस्टिमेशन करते हैं एक स्पेशल ट्यूब में कलेक्ट करते हैं. हमारे पास थी वो ट्यूब, एक स्पेशल ट्यूब में कलेक्ट करते हैं जिसको बोलते हैं फ्लूराइड बल्ब. जस्ट वेट. जिसको बोलते फ्लूराइड बल्ब, जिसकी जो कैप रहती है वो ग्रे कलर की रहती, फ्लूराइड बल्ब। उसमें फ्लूराइड डाला हो रहता, जो जब भी सैंपल कलेक्ट करते पेशेंट का तो वो फ्लूराइड बल्ब में कलेक्ट करते, उसमें रहता सोडियम फ्लूराइड एंड पोटेशियम ऑक्सैलेट। जस्ट वेट, दिस इज फ्लूराइड बल्ब, ग्रे कलर की कैप रहती उसमें। इसमें ब्लड कलेक्ट करते, इसमें रहता सोडियम फ्लोराइड पोटेशियम ऑक्सैलेट इन द रेशियो ऑफ 1:3। तो व्हाट इज रोल ऑफ सोडियम फ्लोराइड? सोडियम फ्लोराइड इनिबिट दिस इनोलेज एंजाइम ऑफ ग्लाइकोलिसिस। Inolase causes the conversion of 2-phosphoglycerate into phosphoenol pyruvate. So this is glycolytic enzyme. This sodium fluoride, this fluoride ion causes the inhibition of inolase by irreversible non-competitive inhibition. It causes by irreversible non-competitive inhibition. This is red cap tube. It is a plain bulb. It is used for the serum analysis. ग्लूकोज छोड़ के बाकी के जितने भी बायोकेमिस्ट्री में पैरामीटर है मैक्सिमम, वो प्लेन बल्ब में हम ब्लड लेते हैं उसका। और जो पैथोलॉजी से रिलेटेड आता है, जहाँ पे सेल मॉर्फोलॉजी देखते हैं, जैसे कंप्लीट ब्लड काउंट है ना, सीबीसी जो करते हैं, वो पर्पल या लेवेंडर कलर की कैप रहती है। ओके, सो दिस इज ईडीटीए ट्यूब, दिस इज प्लेन बल्ब, दिस इज फ्लूराइड बल्ब, दिस इज प्रैक्टिकल एस्पेक्ट। ओके, सो तो ग्लूकोज जब कलेक्ट करते हैं तो ग्रे कलर के कैप में करते हैं फ्लूराइड, क्योंकि ग्लाइकोलिसिस को इनहिबिट करता है। इससे क्या होगा, हमको ट्रू वैल्यू मिलेंगी। अगर हमने अभी ब्लड कलेक्ट किया और 2 घंटे बाद प्रोसेस किया तो भी उसकी वैल्यू मेंटेन रहेंगी बिकॉज ऑफ इनहिबिशन ऑफ ग्लाइकोलिसिस। अगर हमने अभी कलेक्ट किया और 4 घंटे बाद प्रोसेस किया तो द ब्लड ग्लूकोज लेवल डिक्रीजेस टू 5 to 10 milligram पर डेसीलीटर पर आवर। इतने इससे शुगर की वैल्यू कम होते जाएगी बिकॉज ऑफ कंटिन्यूअस ग्लाइकोलिसिस, अगर हमने इसमें कलेक्ट नहीं किया तो। इसलिए ये बल्ब में कलेक्ट करते हैं। दिस इज ऑल्सो ए वाइवा क्वेश्चन। तो इनोलेज, इट इनहिबिट द एंजाइम इनोलेज। इसके इसके बावजूद भी 2 और इनहिबिटर्स है ग्लाइकोलिसिस के, वन इज आयोडो एसिटेट एंड सेकंड वन इज द आर्सिनेट। आयोडो एसिटेट and arsenate, these are the inhibitors of glycolysis. Now the C, point number 24, okay, rate limiting enzyme of glycolysis, ये तो सबको आना चाहिए। rate limiting enzyme of glycolysis, which one is the rate limiting enzyme of glycolysis? Phosphofructokinase is a rate limiting enzyme in the glycolysis, phosphofructokinase. Okay, so what are the important points related with the glycolysis? It has 3 irreversible steps. Glycolysis, it has 3 irreversible steps. 2 substrate level phosphorylation is there. One is 1,3-bisphosphoglycerate kinase, another one is pyruvate kinase reaction. So 2 substrate level phosphorylation reaction is there in glycolysis. 3 inhibitors, okay, अभी बोला iodoacetate, arsenate and the fluoride. Then reversible kinase reaction, काइनेज रिएक्शन जैसे आपने देखा ग्लूकोकाइनेज वाली, हेक्सोकाइनेज वाली रिएक्शन, या फॉस्फोफ्रुक्टोकाइनेज वाली रिएक्शन, या पायरुवेट काइनेज वाली रिएक्शन, जितने भी काइनेज वाले रिएक्शन रहते वो सब इरिवर्सिबल रहते। लेकिन ये अकेली ऐसी रिएक्शन है 1,3-बिस्फॉस्फोग्लिसरेट काइनेज जो रिवर्सिबल काइनेज रिएक्शन है। ओके, टोटल एटीपी प्रोड्यूस्ड इन द ग्लाइकोलिसिस आर via the anaerobic glycolysis 2 ATPs are produced, means glucose is converted into lactate. When under the aerobic condition 7 ATPs are produced, and when it is from the glycogen molecule, when glycolysis is from the glycogen molecule, where you will get glucose 6 phosphate, and from glucose 6 phosphate you will get 3 ATPs because first step is bypassed. So 3 ATPs from the glycogen in the glycolysis. And the point number 25th, this is important point. Citrate is the inhibitor of phosphofructokinase and fructose 2,6-bisphosphate. This compound is a activator of phosphofructokinase. So citrate is the inhibitor, citrate, ATP, these are the inhibitors, while fructose 2,6-bisphosphate, it is a activator of phosphofructokinase enzyme. 2,3-BPG is synthesized in which of the following pathway? So here answer is Rappaport-Lubering cycle. Rappaport-Lubering cycle. This is a Rappaport-Lubering cycle. This is a glycolysis. See, glucose is converted into through various steps it is converted into 1,3-bisphosphoglycerate, and this is converted into pyruvate. This is glycolysis. There is alternate route for the glycolysis in which there is generation of 2,3-bisphosphoglycerate occur, that is known as Rappaport-Lubering cycle. What is significance of this cycle? Because there is generation of 2,3-bisphosphoglycerate, and this compound is very, very important in the RBCs. It is very, very important for in the RBCs because uh, there are three pathways which are important in the RBCs. First one is the glycolysis, second one is the Rappaport-Lubering cycle, and also HMP shunt. These are the three important pathways in the RBCs. Now 2,3-BPG, it decreases the affinity of oxygen with hemoglobin, and it causes unloading of oxygen to the tissues, release of oxygen to the tissues. That's why it is important in the condition like anemia, high altitude, COPD, and in case of fetal hemoglobin, it is very, very important. 2,3-bisphosphoglycerate. This molecule occupy the central cavity of the hemoglobin and causes the unloading of oxygen to the tissue. It causes the dissociation. It causes the shift of oxygen dissociation curve to the right. It causes shift of oxygen dissociation curve to the right. So these are the compound, or these are the negative effectors which causes unloading of oxygen to the tissues or shift the oxygen dissociation curve to the right. So see, increase in the H ion concentration or decrease in the pH, increase CO2, increase 2,3-BPG bisphosphoglycerate, and increase in the temperature. These are the negative effector which shift the dissociation curve to the right. Okay. This is MCQ. This is important point. So curve shifted to the right. Now see the pyruvate dehydrogenase complex. All of you should know that pyruvate dehydrogenase complex is a multi-enzyme complex. It is composed of 3 enzymes and 5 coenzymes. Pyruvate dehydrogenase complex, multi-enzyme complex composed of 3 enzymes and 5 coenzymes. 3 enzymes कौन से है इसमें और 5 coenzymes. These are the 5 coenzymes B1, B2, B3, B5 and lipoic acid. So in other words, that is thiamine pyrophosphate, FAD, NAD, coenzyme A, and lipomide. These are the 5 coenzymes required for the pyruvate dehydrogenase complex, and 3 enzymes: pyruvate dehydrogenase, second one is dihydrolipoyl transacetylase, and dihydrolipoyl dehydrogenase. These are the 3 enzymes. So 3 enzymes and 5 coenzymes are required for the linked reaction which connect the pyruvate to the acetyl coenzyme A, or causes the conversion of pyruvate to acetyl coenzyme A and connect the glycolysis and TCA cycle. Okay, so here answer is pyruvate dehydrogenase. E1 is pyruvate dehydrogenase, E2 is dihydrolipoyl transacetylase, second E2. Okay, और ऐसे और दो एंजाइम है, वन इज अल्फा कीटोग्लूटारेट डिहाइड्रोजनेस एंड सेकंड वन इज द ब्रांच चेन कीटो एसिड डिहाइड्रोजनेस. सो पायरुवेट डिहाइड्रोजनेस जैसे दो और है, दैट इज अल्फा कीटोग्लूटारेट डिहाइड्रोजनेस एंड ब्रांच चेन कीटो एसिड डिहाइड्रोजनेस. सेम इसके जैसे ही है, सिर्फ इस यहाँ पे पायरुवेट डिहाइड्रोजनेस पहला है, वहाँ पे अल्फा यहाँ पे ये ब्रांचन कीटो एसिड डिहाइड्रोजनेज में ब्रांचन कीटो एसिड कार्बोक्सिलेज इज द फर्स्ट वन. 3 enzymes, 5 coenzymes. Point number 29. We'll see the time first. Okay. So see, amino acid linking Krebs cycle and urea cycle. So in Krebs cycle, there is generation of ATP, oxaloacetic acid and CO2. And urea cycle, there is generation of fumarate. How these two cycles are linked? Here answer is aspartate and fumarate. These both are linked with they are linked with the cycle. But here asking amino acid. Here they ask about the amino acids. Okay, see, so Oxaloacetate is converted into aspartate. Aspartate enters the urea cycle, and there is liberation of fumarate, which is used in the TCA cycle. So ATP, they are produced in the TCA cycle, utilized in the urea cycle. CO2, it is produced in the TCA cycle, it is utilized in the urea cycle. Got it? So these are the connecting link between these two cycles. So ammonia, how ammonia depletes, how ammonia causes or affect the TCA cycle. Whenever there is increase in the ammonia, it causes the conversion of alpha ketoglutarate and depletes or decreases the pool of alpha ketoglutarate. So if there is decrease in the alpha ketoglutarate, there is decrease or hamper TCA cycle. Okay, affect the TCA cycle and ATP production. So this is how ammonia causes depletion of alpha ketoglutarate, and it affect the TCA cycle, it affect the ATP synthesis. That's why ammonia is toxic to the brain. Also, it causes the synthesis of glutamine. Increased level of glutamine causes cerebral edema. Okay. So this is point number 30. 31, point number 31. There are 5 disorders which are related with the enzymes of the urea cycle. Okay, so this is hyperammonemia type 1, type 2. This is citrullinemia, arginosuccinate aciduria, and hyperargininemia. These are the 5 disorder, one syndrome, that is triple H syndrome. So these are the 5 disorders: hyperammonemia type 1, type 2, citrullinemia, arginosuccinic aciduria, argininemia. These are the 5 disorders, and one syndrome is there, that is triple H syndrome. Triple H syndrome that indicates increased NH3, hyperammonemia, hyperornithinemia, and homocitrulinuria. Triple H syndrome. It is due to the defect in ORNT1 gene, ornithine transporter gene. Due to defect in this gene, there is failure of transport of ornithine from cytosol to mitochondria. So there is triple H syndrome. So these are the disorders, these are the defective enzymes. Only remember this, there is excretion of orotic acid in type 2 hyperammonemia, because it leads to the increased carbamyl phosphate level, which is diverted to the pyrimidine nucleotide synthesis. And in pyrimidine nucleotide synthesis, there is orotic acid production. Okay, so this is urea cycle. This is urea cycle disorder. Okay, now these are the urea cycle disorders. Now see, this is point number 32. Polysterized, when we already discussed this point, polysterized consumption, because thiamine is present in outer alluron layer of the cereals. It is present in outer alluron layer of the cereals. And polishing मतलब क्या होता है, ऊपर का कवर निकाल डालते घिस घिस के. So that causes the loss of thiamine activity. So 80% of thiamine lost. Okay, thiamine is responsible for oxidative decarboxylation, जो कि 3 हमने एग्जांपल देखे अभी ऑक्सीडेटिव डिकार्बोक्सिलेशन के. कौन से? पायरुवेट डिहाइड्रोजनेज कॉम्प्लेक्स, अल्फा कीटोग्लूटारेट डिहाइड्रोजनेज कॉम्प्लेक्स, एंड ब्रांच चेन कीटो एसिड डिहाइड्रोजनेज कॉम्प्लेक्स. So oxidative decarboxylation and transketolase activity, this is HMP shunt, this is related with the HMP shunt. So this is a function of thiamine. Okay, and in chronic alcoholic, there is decreased absorption of thiamine and decreased conversion of thiamine to thiamine pyrophosphate, that causes Wernicke's encephalopathy and Korsakoff psychosis. Okay, so see, this is polysterized consumption causes thiamine deficiency, means beriberi, and when you eat maize and jowar as a staple diet, it causes the niacin deficiency. Then 33, thiamine, related with the thiamine, there is beriberi. Deficiency of thiamine causes the beriberi. Dry, whenever there is neuropathy, it is known as dry beriberi. When it affects the heart, and when it causes the effusions in the cavity, means accumulation of fluids in the cavity, and edema, it is known as wet beriberi. and when it affect the infant, specifically the cardiovascular manifestation, it is known as infantile beriberi. So Wernicke's Korsakoff psychosis, it is a combination of these symptoms, that is GOA MP, this is mnemonic. The global confusion, ophthalmoplegia, ataxia, memory loss, psychosis. ग्लोबल कंफ्यूजन ऑप्थेल्मोप्लेजिया अटेक्सिया, अगर ये तीन है तो वर्निकेज सिंड्रोम, ओके, एंड मेमोरी लॉस एंड साइकोसिस अगर है तो कोर्सैकॉफ साइकोसिस. दिस इज ड्यू टू द consumption of alcohol, high consumption of alcohol or chronic consumption of alcohol, which decreases the absorption of thiamine from the uh, intestine, and also it alter or it affect the conversion of thiamine to thiamine pyrophosphate. Okay, so GOA MP is mnemonic for Wernicke's Korsakoff psychosis. This is point number 33. Okay. See, क्रोनिक अल्कोहलिक, अगर अल्कोहलिक है तो बहुत सारे डिसीजेस है, अल्कोहलिक से रिलेटेड बहुत सारे डिसीजेस है, जैसे कि फैटी लीवर अल्कोहलिक से रिलेटेड है, सिरोसिस ऑफ लीवर अल्कोहलिक से रिलेटेड है, गाउट प्रेसिपिटेट करता है, मतलब अल्कोहल प्रेसिपिटेट करता है गाउट को, देन अल्कोहल प्रेसिपिटेट करता है थायमिन डेफिशिएंसी को, alcohol thiamine deficiency which causes the Wernicke's encephalopathy and Korsakoff psychosis. अभी symptoms देखो, symptoms of confusion, ataxia and sixth nerve palsy means ophthalmoplegia. So whenever there is these symptoms, it is Wernicke's encephalopathy. If there is memory loss and psychosis additional, it is Wernicke's Korsakoff psychosis. Got it? So GOA MP is the mnemonic. Try to remember this. Now see the point number 34. So poor feeding, convulsions and vomiting, burnt sugar. Urine had smell of burnt sugar and increased branch chain amino acid and their keto acid union. ब्रांच चेन अमाइनो एसिड का नाम आया तो ब्रांच चेन अमाइनो एसिड से मोस्ट कॉमन जो डिसीज है जो रिलेटेड है, दैट इज मैपल साइरप यूरिन डिसीज. देर इज स्मेल ऑफ बर्न शुगर इन द यूरिन. देर इज एक्सक्रीशन ऑफ the branch chain amino acid and their respective keto acid in the urine due to the defect in the enzyme. This alpha keto acid dehydrogenase, thiamine pyrophosphate dependent, or it requires 5 coenzymes, okay, like PDH complex. So alpha keto acid dehydrogenase defect, it causes the maple syrup urine disease. It causes the floppy infant. Intellectual disability is there. There is a smell of burnt sugar. There is increased level of this branch chain amino acid and keto acid in the urine. You may get the DNPH test and Rothra's test positive on the urine sample. So this is defective valine leucine isoleucine metabolism. It is very easy. These branch chain amino acids are first converted into keto acid by the transaminase. Then this keto acid by the dehydrogenase, it is converted into acyl coenzyme A, and via the acyl coenzyme A dehydrogenase, they are degraded into their corresponding product. All of you know that leucine is only ketogenic, so leucine will give you fat. Isoleucine is both glucogenic and ketogenic, so it will give you glucose and fat, and valine will only give you glucose. Okay. So this is point number 34. Here answer is maple syrup urine disease. Black urine in infancy is a diagnostic feature of black urine. It is alkaptonuria. There is deposition of alkaptone, black pigment. यूरिन टर्न्स डार्क, थोड़ी देर रख दिया, जनरली नोटिस नहीं होता क्योंकि इसको टर्न होने को टाइम लगता है. So that is known as alkaptonuria. This is an intermediate step in the phenylalanine and tyrosine metabolism in which homogenticide is converted into 4-mallyl acetoacetate. So for this conversion, homogenticide oxidase is required. If this reaction is blocked, this homogenticide is diverted for the production of benzoquinone acetate. And on polymerization, this benzoquinone acetate will give alkaptone, and deposition of alkaptone in the various tissue, particularly cartilage, ear cartilage, nose cartilage, forehead. The deposition causes the condition that is known as uchronosis. Also, this over a long period of time, there is deposition of this pigment in the intervertebral disc and many bones, causes the, or many joints. It causes arthritis in the adulthood. So it causes arthritis in the adulthood. As such, it is a asymptomatic condition, goes unnoticed in the children. Okay, यूरिन टर्न्स डार्क ऑन स्टैंडिंग, थोड़ी देर रखोगे तो ब्लैक हो जाएगा. ओके, लेकिन वो अननोटिस चला जाता है इसे. सो इट इज, दूसरा कोई भी सिम्टम नहीं रहता, लेकिन ओवर द पीरियड ऑफ टाइम ड्यू टू द डिपोजिशन ऑफ अल्कैप्टोन देयर इज उक्रोनोसिस, द ब्लैक डिपोजिशन, ब्लैक पिगमेंट डिपोजिशन. सो दिस इज अल्कैप्टोन यूरिया. In alkaptonuria, the ferric chloride test, silver nitrate test, these are the positive tests. Okay. Urine turn black on standing. So clinical features of alkaptonuria, point number 36, that is urine turn black on standing, black urine, and main arthritis, tendency to develop arthritis in the adulthood. Test, that is ferric chloride test, is positive, and the silver nitrate test is positive. Silver nitrate test is positive. Also the reducing test for reducing substances is also positive. That is the alkaptonuria. This is again a alkaptonuria, and the another name for the homogentisate oxidase is dihydroxyphenyl acetate dioxygenase. Dihydroxyphenyl acetate dioxygenase. This is another name for the homogentisate oxidase. This is point number. Now the point number 37. Okay, so it is very difficult to cover the 100 points. So we are now on the point number 37. Okay, see, hereditary fructose intolerance. When started winning with the fruit juice contain the fructose. It contains fructose. When the fructose rich food is given to the child, the vomiting, jaundice, hepatomegaly, this type of symptoms are seen. So this is typically a case of there are the severe symptoms. So it is a case of hereditary fructose intolerance due to the defect in the enzyme aldolase B. So aldolase B causes the conversion of fructose 1-phosphate into dihydroxyacetone and glyceraldehyde 3-phosphate, and accumulation of this product fructose 1-phosphate causes this manifestation, that is jaundice, hepatomegaly, also hypoglycemia, hereditary fructose intolerance. Okay. Now the galactosemia, point number 38. Galactosemia, it is due to the defect in the enzyme galactose 1-phosphate uridyl transferase. This is a defect in galactosemia. Okay, due to which there is accumulation of galactose 1-phosphate which causes the manifestation. Also galactose is converted into, this is point number 38, that is galactose 1-phosphate uridyl transferase. There are three types of galactosemia. One is the classical galactosemia, that is due to this defect, and this is minor defect, galactokinase and galactokinase epimerase. These are the minor defect. This is not related with the galactose metabolism, it is related with the gluconeogenesis, also glycogenolysis. This enzyme is defective in the von Gog's disease. So these are the causes of galactosemia, and this is classical galactosemia. And one typical feature of galactosemia is cataract. Congenital cataract. It is due to accumulation of dulcitol. As this enzyme is defective, the galactose is diverted for the production of dulcitol. So dulcitol by the osmotic effect causes the cataract in the lens. Via the osmotic effect, it causes cataract in the lens. So galactosemia, congenital cataract, and the features hepatomegaly, hypoglycemia, and the increase in the unconjugated bilirubin, these all symptoms are due to accumulation of galactose 1-phosphate. Okay. See, jaundice with liver enlargement and eyes shows cataract, and the reducing sugar, test for the reducing sugar, Benedict test is positive, but blood test but the blood glucose estimated by the glucose oxidase method, which estimate the beta D glucose. So test for the glucose in the blood is negative, but the reducing sugar is positive. So that is positive Benedict test, but the GOD POD test is negative. That indicates that it is a case of galactosemia. There is excretion of reducing sugar in the urine. This is point number 40. So this is galactosemia due to defect in the enzyme galactose 1 phosphate uridyl transferase. Now 41, ariboflavinosis. Due to the B12 deficiency. B12 is an important and it is an important vitamin, and it is deficiency of B12 नहीं B2 राइबोफ्लेविन और राइबोफ्लेविनोसिन फ्लेविनोसिस. अगर इसका डिफेक्ट हो गया, अगर इसकी डेफिशिएंसी हो गई, इट कॉजेज द एंगुलर स्टोमेटाइटिस, ग्लोसाइटिस, चिलोसिस, seborrheic dermatitis, conjunctivitis like feature. Circumoral features are there. So this is due to vitamin B2 deficiency, riboflavin deficiency, that is known as ariboflavinosis. Okay, FAD. So this is 41. Now the 42. Vitamin K deficiency. Vitamin K for coagulation. It is required for the clotting. The supplementation will help a neonate suffering from. It is for the hemorrhagic diseases. It is important for the hemorrhagic diseases. K for coagulation. Vitamin K causes post-translational gamma carboxylation. Translational gamma carboxylation of clotting factor 2, 7, 9 and 10. Clotting factor 2, 7, 9 and 10, and it is responsible for the clotting process. So it prevents the hemorrhagic disease. So menadione injection is given at the birth to prevent the bleeding episodes. Okay, so the hemorrhagic disease. 43, 44, 45. This is very important. Gaucher's disease, the top rated MCQ for any 5-star MCQ for any examination, whether FMGE or PG entrance examination. Okay, Gaucher's disease. So in the Gaucher's disease, it is due to the defect in the enzyme beta glucocerebrosidase. Beta glucocerebrosidase. Okay. So see, fatty acid plus pingosine. It is known as ceramide. This is ceramide. This is important point. This is point number 43. It is ceramide. If ceramide plus glucose, it is known as glucosyl ceramide or glucocerebroside. If the defect is there, glucocerebrosidase defect is there, this glucocerebroside, it accumulates particularly in the liver, spleen and bone marrow. Liver and spleen. It causes hepatosplenomegaly. In the liver and spleen. And in bone marrow, it causes avascular necrosis of the bone. Avascular necrosis, osteoporosis, frequent fractures. These are the features. So these are the features of the Gaucher's disease. This is the most common lysosomal storage disorder. And this is one point. Then deficiency of glucocerebrosidase, that is beta glucosidase defect, it causes Gaucher's disease. And the typical features, it gives the crumpled tissue paper appearance, crumpled paper or tissue paper appearance of the lysosomes on microscopy. So this is a feature or wrinkled paper or crumpled tissue paper appearance of the lysosomes in the microscopy, and it gives the Erlenmeyer flask deformity. This is Erlenmeyer flask, and if you see the femur, the end of the long bone, it appears like Erlenmeyer flask on the X-ray. Erlenmeyer flask deformity on X-ray. These are the features of the Gaucher's disease. Apart from this, the point number 46, this is very, very important. Apart from these points, there is no cherry red spot in the Gaucher disease. No cherry red spot on macula. There is no cherry red spot in KGF, Krabbe's disease, Gaucher's disease, and Fabry's disease. Krabbe's, Gaucher, and Fabry's disease, there is no cherry red spot on the macula. So these are the features of the Gaucher's disease because of deficiency of beta glucocerebrosidase. So these are the disorders. इसमें से कौन से ध्यान में रखने के हैं? This is Gaucher's. It is most common. Affect the bone, liver, and spleen. There is hepatosplenomegaly. There is no cherry red spot, and it affects the bone. So avascular necrosis, osteoporosis, frequent fractures of the bone, and the wrinkled tissue or crumpled tissue paper appearance. Beta glucosidase is the defective, and glucocerebroside accumulation. Second is the Niemann-Pick disease. It's due to sphingomyelinase deficiency, and there is hepatosplenomegaly. In this disease, also hepatosplenomegaly, it has cherry red spot on the macula, and it is due to sphingomyelin accumulation. The next important is the Tay-Sachs disease. There is cherry red spot on the macula due to the defect in hexosaminidase A, and there is no organ involvement. No organ involvement. It is GM2 gangliosidosis. So these are the important disorders, lysosomal storage disorders. So point number 47, this is point number 47. This is point number 48. Then point number 49, that is no cherry red spot in KGF. Okay, point number 49. Okay, so these are the important disorders. Of this, wait, point number 50, Fabry's disease. Fabry disease. This is X-linked. All are autosomal recessive. This is X-linked disorder because of alpha galactosidase defect, and there is angiokeratomas, telangiectatic skin lesion in the Fabry's disease. This is point number 15. So overall lysosomal storage disorder, these are the frequently asked MCQs, but Gaucher's is the most commonly most commonly asked disease. Gaucher's disease. Crumpled tissue paper microscopic appearance. Okay, Gaucher's disease. See, this is crumpled tissue paper appearance because of defect in the glucocerebrosidase, beta glucocerebrosidase. No organomegaly. See, if you are suspecting the lysosomal storage disorder with no organomegaly, that is GM2 gangliosidosis, this is Tay-Sachs disease. Here answer is Tay-Sachs disease. In Gaucher's disease, there is hepatosplenomegaly. Niemann-Pick disease, this is hepatosplenomegaly. And Hunter's disease, there is hepatosplenomegaly. On examination, cherry red spot is there. So cherry red spot is there in the Niemann-Pick disease, in the Tay-Sachs disease, but no, not in the Gaucher's and Hunter's disease. Okay, so this is important. Okay. We already discussed this point. Which of the following is considered as universal methyl donor? It is SAM. This is sulfate donor. Universal sulfate donor, specifically required for the glycosaminoglycan synthesis. Okay. So this is very, very important now. The source of glucose. Okay. Now see here, answer is liver, glycogenolysis. So person had a meal at 8 p.m. and record blood glucose at 7 a.m. in the morning. So total 11-hour gap is there. Total gap is 11 hours. Now see, dietary glucose will supply the glucose for initial 2 to 4 hours after the meal. Initially 2 to 4 hours after the meal, it is supplied by the energy is supplied by the dietary glucose. Okay. Then after this, the glycogenolysis is the important pathway. From 4 hours to near about 16 hours, the glycogenolysis or glycogen breakdown in the liver is important source. Muscle will not give you glucose because there is no glucose 6 phosphatase enzyme in the muscle. There is no glucose 6 phosphatase enzyme in the muscle. So muscle glycogenolysis will not provide you glucose. There is glycolysis, but will not provide you glucose. Glycolysis will start from the glucose 6 phosphate. Okay, so hepatic glycogenolysis will provide you the glucose for 4 to 16 hour. Then after 12 hours, means after 16 hour, 18 hour to 48 hour, this is taken care by the gluconeogenesis. Taken care by the gluconeogenesis, and during the prolonged starvation, means prolonged fasting, or during the early starvation, then the beta oxidation, ketone body metabolism, they will take over, and the last stage, the proteolysis, muscle proteolysis is important. So this is important point number 51. Point number 51. Now this is a case of iron deficiency anemia. See, decrease hemoglobin, ferritin, and mean corpuscular volume. So see the features: excessive tiredness, poor appetite, inability to concentrate, tingling sensation, pallor. Okay, these are the features of iron deficiency anemia. Okay, zinc deficiency, point number 52. Zinc deficiency causes acrodermatitis, enteropathica. Dermatitis typically involving the acral parts of the body, mouth, nose, fingers, buttock. They are particularly involved in the zinc deficiency. Also, there is delay in the wound healing. There is loss of taste sensation, and alopecia is there in the zinc deficiency. Copper deficiency causes, point number 53, copper deficiency causes Wilson's disease. It's not copper deficiency, but copper metabolism defect, Wilson's disease, and Menkes disease. Menkes disease. These are not features of Wilson's or Menkes disease. It also causes the anemia, but it is less severe as compared to the iron. Calcium deficiency causes tetany. Tetany means carpopedal spasm. Carpopedal spasm. And the iron deficiency causes iron deficiency anemia. It causes iron deficiency anemia, in which there is decrease in the hemoglobin level decreases, ferritin level decreases, the MCV, it is decreases or less MCV, mean corpuscular volume, but TIBC, total iron binding capacity, will increase in the iron deficiency anemia. Iron is a one-way substance. There is mucosal block theory in the iron absorption. Point number 56. Iron is absorbed by the divalent metal ion transporter 1. From the intestinal surface, it is absorbed by the divalent metal ion transporter 1. This is particularly important for absorption of the divalent metal, particularly lead, zinc, copper, and the iron. It is important. Ferrous, in the ferrous state. Okay. Then hepcidin is the protein which is important for the regulation of iron absorption. It particularly if the iron levels are more, it decreases the divalent metal ion transporter and ferroportin levels. Hepcidin regulate the iron level in the body. Hepcidin regulate the iron level in the body. It decreases the DMT and decreases the ferroportin level. when iron levels are high and regulate the iron level. There are number of iron related proteins. You should know that, and you should know the sources of the iron. You should know the number of iron related protein. I will enumerate one by one. So iron is present in hemoglobin, myoglobin, cytochrome P450, catalase, tryptophan pyrrolase, nitric oxide synthase. फिर से hemoglobin, myoglobin, cytochromes, cytochrome P450, tryptophan pyrrolase, catalase, xanthine oxidase. Then again it is present in the ferritin, transferrin, hemosiderin. Ferritin, transferrin, hemosiderin, it is present. Got it? So iron, it is important in the multiple enzymes. It is present. Okay. In multiple enzymes, iron is present, and that particularly include hemoglobin, myoglobin, cytochrome P450, tryptophan pyrrolase, xanthine oxidase, then nitric oxide synthase that contain iron. Iron is an important micro element. It is a trace element. When the requirement is less than 100 milligram, it is known as trace element. It belongs to. It is a one-way substance. One-way substance. Deficiency causes hypochromic microcytic anemia. Deficiency of iron causes hypochromic microcytic anemia. The rich source of iron is green leafy vegetable, where the iron content is much more. Mainly the green leafy vegetable spinach. Okay, spinach. After spinach and all other sources, then comes the meat. Okay, so the green leafy vegetable, these are the important sources for the iron. So all the points related with the iron, they are very, very important. If there is iron deficiency, there is increase in the TIBC, total iron binding capacity. There is decrease in the hemoglobin, ferritin, and MCV. Whenever there is increased demand, there is iron deficiency anemia, or whenever there is increased blood loss, there is iron deficiency anemia. Okay, so these are the important points related with the iron. Okay. So see, child presented with history of rashes around the eyes and mouth, impaired night vision impaired in the vitamin A deficiency. Okay, but here no, it is not about the vitamin deficiency question. Okay, the wound healing is not proper. So wound healing, point number 61, wound healing, impaired wound healing, it is seen in vitamin C deficiency, vitamin C deficiency, then it is seen in the zinc deficiency and hyaluronic acid deficiency. Delayed wound healing, वुंड हीलिंग से रिलेटेड विटामिन सी है, जिंक है और हाइलोरोनिक एसिड है. जिंक के फीचर मैंने आपको बताए थे. जिंक के कौन कौन से फीचर बताए थे. अगर जिंक डिफिशियंसी है तो इट कॉजेज द डर्मेटाइटिस पर्टिकुलरली इन्वॉल्विंग द एक्रल पार्ट ऑफ द बॉडी, मतलब finger, nose, mouth, buttock, these parts are involved. Then loss of taste sensation, because zinc is present in the gustine protein which is present in the taste buds. So loss of taste sensation. Then spermatogenesis is also affected. Then it causes alopecia and delayed wound healing. These are the symptoms of zinc deficiency. Okay, 61. Now the point number 62. This is very, very important. It is an coenzyme in more than 300 enzymes, cofactor. It is an cofactor in more than 300 enzymes, particularly the carbonic anhydrase, carboxypeptidase. These are the important one. Carboxypeptidase, then superoxide dismutase, then alcohol dehydrogenase, then lactate dehydrogenase, and the RNA polymerase, protein synthesis. These are the important enzyme in which zinc is an integral component. So more than 300 enzymes. So carbonic anhydrase, it is zinc. Lysyl oxidase, it is copper related. Lysyl oxidase, particularly it is important for the cross linking in the elastin molecule, and it is blood vessel, it is important. If it is defective, it causes the aneurysms. Okay. Carboxylase, it is biotin dependent. Kinase magnesium dependent. Okay. So these are the important. So this is point 62. Okay. Also important point, that is 63. It is required for the release of insulin. Release of insulin from the beta cells of the pancreas. So it is important for the release of insulin, but it does not potentiate the action of insulin. For the action of insulin, it is potentiated by chromium. Chromium. So it is known as glucose tolerance factor. Glucose tolerance factor. It potentiates the action or increases or potentiates the action of insulin. So it is known as glucose tolerance factor, chromium. Point number 64. वन बाय वन पॉइंट आप लोग नोट करो. ठीक है. Zinc, it causes acrodermatitis enteropathica, delayed wound healing if there is deficiency, loss of taste sensation, spermatogenesis is affected. It causes the release of insulin, so it is also defective. It is an integral component of more than 300 enzymes. These are the important ones. Chromium is also known as glucose tolerance factor, and it increases or potentiate the action of insulin. Okay, this is about the zinc. Now see, KF ring, Kaiser-Fleischer-Ring, golden brown colored ring around the cornea in the decimate membrane of the cornea. Okay, so it is seen in Wilson's disease. 65, KF ring, golden brown ring around the cornea. It is seen in the Wilson's disease because of copper deposition. Okay, in Menkes disease, there is Menkes kinky hair. The brittle hairs are there in the Menkes disease along with the hypopigmented patches. Acrodermatitis, this is zinc related. Tetany, it is calcium related problem. Okay, Menkes and Wilson, these are the copper related. Okay, copper. Reported with jaundice, fatigue, tremor and behavioral changes. Okay, cerebrohepatorenal disease. Hepatolenticular degeneration. Wilson's disease is also known as hepatolenticular degeneration. Hepatolenticular degeneration. So KF ring is there. See the KF ring in the decimate membrane of the cornea. Wilson's disease. So this is point. Wait. See the 67, 68. You should know about the Menkes disease and Wilson's disease. Menkes disease, it is due to the mutation in the ABC transporter protein, that is ATP7A. What is the role of ATP7A? It is required for the absorption of copper. Absorption of copper from the intestine. So if it is defective, there is copper deficiency that causes the Menkes disease. So specifically the lysyl oxidase is affected in the Menkes disease. There is CVS malformation, there is brittle hair in the Menkes disease and hypopigmented patches. It is a X-linked disease. Typically affect the males. Menkes disease. Okay. And the ATP7B mutation causes the Wilson's disease, and it is required for excretion of copper into the bile. तो अगर ये डिफेक्टिव है तो कॉपर एक्यूमुलेट हो जाएगा एंड इनकॉर्पोरेशन ऑफ कॉपर इनटू द सेरुलोप्लाज्मिन. सो सेरुलोप्लाज्मिन लेवल विल बी decrease सेरुलोप्लाज्मिन in the Wilson's disease. Copper is excess, but ceruloplasmin levels are low because incorporation of the copper into the ceruloplasmin is low. Also, excretion is low. There is copper excess in the Wilson's disease, which causes KF ring in the cornea, and hepatolenticular liver and lenticular nucleus of the brain is involved. and there is also the lens is also involved. Okay, but here lenticular means lenticular nucleus of the brain. So 69 hormone that regulate the calcium levels. So calcitonin, vitamin D, and parathyroid hormone, they regulate the calcium level. The vitamin D increases the calcium level. The parathyroid hormone increases the calcium level, while calcitonin decreases the calcium level. They particularly act on the three target tissue, that is bone, intestine, and renal tubules. Renal tubules. Calcium deficiency causes tetany, that is carpopedal spasm, in which this is an emergency condition in which calcium gluconate is given. The Chostek sign means tapping on the facial muscle causes twitching, facial twitching. So that is known as Chostek sign. And Trousius sign means if you inflate the BP cuff more than systolic blood pressure, means up to the 200, it causes the carpopedal spasm. So that is Trousius sign. So Chostek and Trousius signs, they are positive. And carpopedal spasm, this is a typical position of hand. Flexion of the wrist, extension of the interphalangeal joint, and flexion of the metacarpophalangeal joint. So this is carpopedal spasm in the calcium. It is a major element, calcium. Also clotting factor 4. Now the important point number 70. Point number 70. Glutathione peroxidase contain which metal? The selenium, it is important in 3 enzymes. One is glutathione peroxidase. Second one is the deiodinase. And third one is the thioredoxin reductase. One is glutathione peroxidase. So this is important. Selenium, it is important in the glutathione peroxidase. Okay. Cation disease. Cation disease, it is due to deficiency of the selenium. It causes cardiomyopathy or cardiac involvement in the selenium. Zinc deficiencies causes acrodermatitis enteropathica. Copper deficiency causes Menkes disease. Copper excess causes Wilson's disease. Iron deficiency causes iron deficiency anemia, while excess causes hemosiderosis, hemochromatosis. Selenium deficiency causes the Cation's disease. This is 71. Selenium deficiency causes Cation's disease. 72. Micro element, when the daily requirement is less than 100 milligram per day, they are known as micro element. When it is more than 100 milligram per day, it is known as major elements. So calcium requirement is near about 500 to 800 milligram per day, near about 800 milligram per day. Pregnancy and lactation again 1200, 1500 milligram per day. So calcium is the major element. Okay. So copper, it is near about 2 milligram. Selenium, it is in microgram. Zinc, it is in near about 10 milligram per day. So calcium is the major element here. Easy bruising and bleeding gums. This is very easy. It is related with vitamin C. Scurvy. Okay. So petechial hemorrhages are there. Gum bleedings are there because of defective collagen cross-linking or defective collagen synthesis. Vitamin C, it is required for the hydroxylation reaction. And deficiency of vitamin C, it affects the hydroxylation of proline and lysine and defective collagen synthesis, which causes easy bruising and the petechial hemorrhages. So this is scurvy, vitamin C deficiency. 74. Vitamin K is required for the coagulation. Is there any option coagulation? No. So it is required for the कार्बोक्सिलेशन, पोस्ट ट्रांसलेशनल गैमा कार्बोक्सिलेशन. कार्बोक्सिलेशन के लिए बायोटिन लगता है, ट्रांसअमाइनेशन के लिए पायरिडोक्सिल फॉस्फेट लगता है, हाइड्रोक्सिलेशन के लिए विटामिन सी लगता है. एंड सम रिएक्शन, सम कार्बोक्सिलेशन रिएक्शंस दे आर बायोटिन इंडिपेंडेंट. उसमें से एक है विटामिन के डिपेंडेंट गैमा कार्बोक्सिलेशन विच इज ए बायोटिन इंडिपेंडेंट कार्बोक्सिलेशन रिएक्शन. ऐसे ऐसे 4 रिएक्शन है जो बायोटिन इंडिपेंडेंट कार्बोक्सिलेशन रिएक्शन है. कौन से 4 रिएक्शन है? बायोटिन इंडिपेंडेंट, बायोटिन इज 75, बायोटिन इंडिपेंडेंट carboxylation, CO2 fixation. Biotin independent carboxylation reactions are, the first one is the CPS1, that is urea cycle, CPS2 pyrimidine nucleotide synthesis, then AIR carboxylase purine nucleotide synthesis reaction, AIR carboxylase, and fourth one is the gamma carboxylation. These are the biotin independent carboxylation reaction. तो gamma carboxylation के लिए क्या लगता है? विटामिन के लगता है. ठीक. Which of the following helps in wound healing? wound healing के लिए हमने ऑलरेडी डिस्कस किया था, विटामिन सी, जिंक एंड द हाइलोरोनिक एसिड. तो दिस इज हाइलोरोनिक एसिड which is required for the wound healing. Then 76, hyaluronic acid, the GAG, 76 glycosaminoglycan. It is very, very important. Each and every point related with the glycosaminoglycan is important. Point number 76. Look, please. Okay. We'll conclude in the 10 minute. Okay. So glycosaminoglycans, they are important. Okay. So glycosaminoglycan not containing uronic acid is keratan sulfate, does not contain uronic acid. Glycosaminoglycan with L-iduronic acid is dermatan sulfate. Okay. Then distribution of glycosaminoglycan, So hyaluronic acid in the synovial fluid, vitreous humor of the eye, then heparin in the lung, in the blood vessels, in the liver, then the heparin that is chondroitin sulfate in the cartilage. It is responsible for compressibility of the cartilage. Then keratan sulfate in the cornea. Keratan sulfate, it is responsible for transparency of the cornea. Transparency of the cornea. Okay. And the Hurler syndrome, Hunter syndrome, various mucopolysaccharidosis, they are related with the glycosaminoglycan. The acid-base disorders. So acid-base disorders we'll discuss here. Wait. How you will diagnose acid-base disorder? Just look for the pH, whether acidic or basic, and this is your primary defect. सबसे पहले pH देखना है. अगर एसिडिक है तो एसिडोसिस, बेसिक है तो दैट इज अल्कोलोसिस. इतना समझ में आया? अगर pH 7.35 के नीचे है तो एसिडोसिस और 7.45 के ऊपर है तो अल्कोलोसिस. नाउ लुक फॉर द बाइकार्बोनेट. इफ इट इज लेस देन 22, बाइकार्बोनेट मतलब बेस है. बेस अगर तो कम हो गया तो एसिडोसिस, बेस अगर कम हो गया तो एसिडोसिस, बेस अगर बढ़ गया तो अल्कोलोसिस. एंड दिस इज मेटाबोलिक. Bicarbonate is the metabolic component. And PCO2, carbonic acid, compare it with the carbonic acid. PCO2, the carbonic acid, अगर PCO2 बढ़ गया तो एसिड बढ़ गया तो एसिडोसिस, PCO2 बढ़ गया तो रेस्पिरेटरी एसिडोसिस, PCO2 कम हो गया तो रेस्पिरेटरी अल्कोलोसिस. तो बाइकार्बोनेट मतलब बेस, बाइकार्बोनेट कम हो गया तो एसिडोसिस, बढ़ गया तो अल्कोलोसिस। CO2 मतलब कार्बोनिक एसिड, कार्बोनिक एसिड बढ़ गया तो एसिडोसिस, कम हो गया तो अल्कोलोसिस। pH 7.35 acidosis, 7 more than 7.45 alkalosis. Okay, these are the important points related with this. Now one important condition, see, this is the case of diabetic ketoacidosis which is frequently asked. Okay, so this is diabetic ketoacidosis, point number 80, diabetic ketoacidosis. So in diabetic ketoacidosis, both Benedict test and Rothera's test, they are positive. So this is Benedict positive, orange red color, that is positive Benedict test, and the purple ring at the junction of two liquid, that is positive Rothera's test. You may get a image based question on this. Okay, so Benedict test, Rothera's test positive means it is a diabetic ketoacidosis. It is a cause for high anion gap metabolic acidosis. Now see the pH is down, means it is acidosis. See for the PCO2, 35 to 45 is the normal. PCO2, it is decrease, means it indicates alkalosis. Okay, and bicarbonate decrease means acidosis. Those who goes with the pH is the primary defect. So here the metabolic component goes with the pH, means it is a primary defect. It is a metabolic acidosis with compensation. Compensation. So metabolic acidosis with compensation is the answer, but there is no option here. So metabolic acidosis is the option. Apart from this, point number 81, in diabetic ketoacidosis, there is increased potassium level, that is hyperkalemia. To compensate the increase H आयन कॉन्सेंट्रेशन, जो एक्स्ट्रा सेलुलर फ्लूइड में या ब्लड में हाइड्रोजन आयन का कॉन्सेंट्रेशन बढ़ा है एसिडोसिस की वजह से, वो कॉम्पेंसेट करने के लिए पोटेशियम सेल के अंदर से बाहर आता है और हाइड्रोजन आयंस जो रहते हैं वो अंदर जाते हैं। टू कॉम्पेंसेट मीन्स इट इज रीडिस्ट्रीब्यूशन, टू कॉम्पेंसेट दैट पोटेशियम बाहर आते तो हाइपर कैलेमिया हो जाता। लेकिन इंसुलिन जब देते हैं तो इंसुलिन ओपन्स द पोटेशियम चैनल एंड दैट सडनली कॉजेज द हाइपोकैलेमिया। दैट्स वाई स्ट्रिक्ट मॉनिटरिंग while giving the insulin in diabetic ketoacidosis is mandatory. Okay, so this is 81. Then 82, the major buffer in extracellular fluid is the bicarbonate buffer, and it is important buffer because of the quantity, and these parameters are under the physiological control. CO2 by the lung and bicarbonate by the kidneys. Okay, and major intracellular buffer is the phosphate buffer. Major intracellular buffer is the phosphate buffer. It is a most potent buffer, and pK is nearer to the blood pH 6.8. That's why it is a more potent buffer in the intracellular environment inside the cell. This is point number 83. So these are the causes of metabolic acidosis, alkalosis, respiratory acidosis, alkalosis. Okay. Now you should be able to differentiate between the three types of jaundice and neonatal physiological jaundice. See, hemolytic, hepatic, we will discuss this table and we will conclude. So hemolytic, hepatic and obstructive jaundice, these are the three important types of jaundice. If the parameters, all parameters are near about normal, all the enzymes SGOT, SGPT, ALP and these obstructive enzymes, if they are normal, it is a hemolytic jaundice. See, there is increase in the indirect bilirubin and urinary urobilinogen is present. This is a classical test, and absent urine bilirubin. These are the features of hemolytic jaundice, particularly the G6PD deficiency, hemolytic jaundice. Okay. Then hepatic jaundice, when these parameters, when this liver enzyme SGOT, SGPT are marked raise near about 10 times or 15 or 20 times of the normal, then it is hepatic jaundice, and there is slight increase in the obstructive enzymes. While in the obstructive jaundice, there is marked increase in these ALP, GGT, 5' nucleotidase, there is marked increase with slight increase in this SGOT and SGPT. Now see, in hepatic jaundice, the Vandenberg reaction is biphasic. That means there is increase in the direct as well as indirect bilirubin. While in the hemolytic jaundice, there is increase in the indirect bilirubin, while in obstructive jaundice, there is mainly increase in the direct bilirubin. In hepatic jaundice, the Vandenberg reaction is biphasic, means there is increase in both indirect and direct bilirubin. Okay, so urinary urobilinogen is absent in the obstructive jaundice. The color of stool, it is pale colored stool in obstructive jaundice, while it is dark color in the hemolytic jaundice. So this is how you can differentiate between the different types of jaundice. So these are the different points related with the FMGE examination. So thank you very much. Keep watching.